So now we're going to start with our lecture of the mRNA splicing, which is the third part of our protein synthesis. So before, we've already talked about transcription, where we used our DNA strand in order to make the mRNA. And we already talked about mRNA editing, where we added on the 5' cap and the 3' poly A tail. Are we good with that?
Yes.
Awesome. Here's the next step that we have to do to protect and get this little mRNA strand ready to go out into the crazy world of the cytoplasm. Because remember, we have to get them out of the nucleus and into the ribosomes in order to make the proteins. On the board here, I'm going to use orange and blue. Here is our mRNA strand. It's going to have different sections in it. That's going to have either orange or blue. And they alternate. Now, the length of these sections can differ depending as to which protein that we're going to want to make. But let's say, for example, that we're going to use this order and this size of our different sections here. You'll notice that we have an orange and then a blue, and then an orange and then a blue and then an orange. The orange we call exons. So exon number 1, exon number 2, and exon number 3. Okay. The blue sections we're going to call intron. Intron 1 and intron 2. Now, the blue sections, through this process of splicing, we're going to get rid of the blue sections. The blue sections do not code for the protein that we're trying to make, so we need to get them out of there. A good way to think about this is that they don't code, and they are in the way.
These are all codons?
These are all like sections of the mRNA strand. So we need to make sure that we have the correct codons that we need to make this specific type of protein. When we copied the DNA, we got some other chunks here that may or may not code for what we need. So we're going to try and get those chunks and remove those chunks. The orange, the exons, do code for the protein. And to remember this, the exons are expressed. The exons are expressed.
What does that mean?
Expressed means that those are going to show up. So if you think of like a trait is something that is being expressed. So either your hair color, your eye color, whether you can roll your tongue, any of that sort of stuff, those are traits that are expressed. Proteins help make up your traits. So this is the area that we're going to code, and that's the area that's going to be expressed either in making a protein, which might help with something in our protein synthesis, it might help in something in our cell membrane, or it might make a trait like this. Okay? Yes?
Can this cause mutations of the
Mutations don't happen as much with this area of it. Mutations happen more with the transcription, with the copying down of stuff. Here we are just kind of taking out this chunk of it, and we're going to stick these exons all together to form one solid piece of mRNA that we're then going to turn into a protein. You guys want to see a video as to how this actually happens?
Yes, yeah. Yes, ma'am.
Thank you. Very polite.
There we go.
Possessing introns and exons.
Okay, so we already have the introns and exons. We only have one enzyme this
Here
time.
we see an RNA molecule with a single intron. Several signals exist within the intron that are used in the splicing process. From the 5' end of the intron, these are GU, the A branch site, a pyrimidine-rich region, and the 3' AG. The GU and AG sequences define the beginning and end of the intron. Splicing is mediated by the spliceosome, which consists of several protein RNA complexes. The first step involves two complexes that bind near the GU sequence. The RNA is then looped, and 3 other protein RNA complexes bind. This final complex then undergoes a conformational change.
Just means that it's changing shape.
Introns are non-coding RNA sequences that must be removed before translation. The process of removing the intron is called splicing. The intron is then cleaved at the 5' GU sequence and forms a lariat at the A branch site. The 3' end of the intron is next cleaved at the AG sequence, and the 2 exons are ligated together. As the spliced mRNA is released from the spliceosome, the intron debranches and is then degraded.
Wonderful.
Watch that again.
We can certainly watch that again. Do you want to watch it again right now? Or after we break it down?
After.
After we break it down. Okay. So let's go through here. I'm actually going to draw these in stages. So this is our first stage here, and then we're going to move over here. So the first thing that they said is that we're going to have a spliceosome kind of attach a couple here. Just where that intron and the exon meet. Okay? It's going to start to fold over. So we have a couple parts of our exons there. And then our intron is kind of the one that's looped around. We're just going to focus on taking out intron number 1 for what's on the board here. This whole process is then going to happen again with intron number 2. So we folded this over here so it's easier to get to. And we're going to have our spliceosome kind of attach on both sides there. Now, the next stage is that we're going to have our intron kind of attach and make a tiny little loop there. So what it did is that it cut right here and then took the end and looped it around and stuck the end onto itself.
That's weird.
So it makes like a little loop. They called it this fancy name called a lariat. It just means that it's like this little loop here. Okay. Then we're going to have... Yep, there's only a couple of steps here. So we have step 1 is we're identifying where the introns and the exons are. Step 2 is that we used the spliceosome and we kind of folded it over and cut that first part. Step 3 is we made this little loop. Then step 4 is we're going to break that little loop off, and we're going to have 2 exons that are not connected to the intron anymore. So we sliced it right in there. So originally we had sliced it here and looped that end around. Next we're going to slice the other end of the intron away from the exon. So this kind of floats away. That's now separated. Okay. The last thing that we do is we want to stick the intron or the exons all back together so that we have one big long chain again.
That's the last phase, just a line.
That's the last phase. So we have exon number 1 is kind of here, and exon number 2 is kind of here. But they're all stuck together. We took that intron out.
So that's a strand of mRNA.
That's a strand of mRNA. And that's the strand of mRNA that we're then going to bring over to the ribosomes in order to make it to our protein. Okay.
What was it before the splicing happened?
Before the splicing happened, it was called an unedited mRNA. So it had the same cap and poly A tail on it, but it wasn't quite done. We had to edit it by taking the introns and removing those guys. Do you guys want to see the video again?
Yes.
What happens to the intron?
The intron, great question. The intron is actually going to be taken and it's going to be broken down and degraded. Remember, this is all happening in the nucleus. So these RNA molecules, the RNA nucleotides, will then get recycled in order to make another strand of mRNA.
Oh, that's sweet.
Yes, ma'am.
Or just reuse recycle.
Words like time and the A distinction or something like that. Do you need to know those? Because you never said them when you were
So the GU that they were talking about is the beginning part here. That's where it's initially going to cut it here.
right. And then they said
Where it bonds is called the A branch. So that's where that little loop connects again. And then there's a final part here where it's going to slice it off. Okay. All right. Let's watch the video one more time, and then I'll show you guys how to identify where the introns are.
To the process of translation. Key factors in this process include RNA, possessing introns and exons, and the spliceosome. Here we see an RNA molecule with a single intron. Several signals exist within the intron that are used in the splicing process. From the 5' end of the intron, these are GU.
Oh. So the GU is going to be right here where we first break it off. That's the first part of that intron.
The A branch site,
That's where we're going to attach the loop of our intron here.
a pyrimidine-rich region, and the 3' AG.
Which is our final breaking point of our intron. Okay.
The GU and AG
AG's on the end.
sequences define the beginning and end of the intron. Splicing is mediated by the spliceosome, which consists of several protein RNA complexes. The first step involves two complexes that bind near the GU sequence. The RNA is then looped, and 3 other protein RNA complexes bind. This final complex then undergoes a conformational change. Introns are non-coding RNA sequences that must be removed before translation. The process of removing the intron is called splicing. The intron is then cleaved at the 5' GU sequence and forms a lariat at the A branch site. The 3' end of the intron is next cleaved at the AG sequence, and the 2 exons are ligated together. As the spliced mRNA is released from the spliceosome, the intron debranches and is then degraded.
That makes so much sense.
Right? Doesn't it make sense now? So all we're doing is we're just making a little loop and taking out this little blue chunk, the intron, and we're going to stick the orange chunks back together. We're going to splice together our exons. Are we good with this? Did I lose anybody?
Yeah.
You have a question?
Yo, comprehendo.
Just say yeah, one thing.
Oh, yeah, that's you're going to move on? Okay, perfect. So now let's talk about how we find an intron. Let's go back to that. I should not have used the green. Let's go back to that practice page that we were working on previously. And on the back side, we have a couple of practice problems for how to do this. I'm going to walk through number 6 with you guys and show you guys how we can find the introns and take them out. And then I'll have you guys practice the last couple of questions on this sheet here. So I'm going to copy this page, or this number 6 up on the board here. U, A, C, A, C, G, C, U, A, G, C, A, C, C, U, G, A. Okay. So this is our unedited strand of mRNA. Yes, Gary?
What's for these again, what's this called up here? They're normal RNA. The way top stuff?
The way top stuff?
Yeah, there's 4 and 5.
Number 4 and 5. That is the DNA. And then you're making the mRNA in order to make the amino acid. With numbers 6, 7, 8, and 9, we are given the mRNA molecule, and we're going to try and find the introns that we're then going to remove. So it does give you the amino acid strain that it's supposed to be making, and that is tyrosine, alanine, serine, proline, and then stop. So stop is not necessarily an amino acid, but it's a code in order to tell the translation process that we're done making this chain. So when we hit the stop, we stop. We don't have to worry about any codons after that, because our process is then done.
Wait, it's just like there's nothing there for stop, like the actual thing?
Correct. It's just a code to say, okay, we're done here. We can break off now.
It just stops.
Yes.
Is there a protein for stop so that like if they try to copy the one they just made with that?
Does it ever turn to a green light?
Stop?
It just kind of signals that it's going to stop, and then we're done with this process. So we're not going to add an amino acid in for that.
I just can't wait for Spanish for Joe.
Right. Amaya?
What's going to happen?
Tia? Thank you.
Yes.
Jaden, yes.
Are the stop parts what tells the
Yep, so when we're doing our translation, when we come back next week and we talk about translation, we'll talk about how we're bringing these amino acids in. We're practicing this first in order to kind of understand how we're bringing all of these pieces together, and then we'll talk about the process after we come back for next week. Okay?
You're going to put the splicing thing up?
Yes, absolutely. Okay. This whole splicing thing is going to go up on the board behind my desk. Okay? Let's find the introns. So we're going to look at UAC, and if you look on the codon wheel, does UAC code for tyrosine?
Yeah.
Yes.
UAC codes for tyrosine. So we got that one correct. We're going to draw a line after that, and I like to draw kind of an arrow to say that's where that one is. Let's look at our next three. Does ACG code for alanine?
No.
No. So then we need to cross off the A and try again. Does CGC code for alanine?
No.
No. So we're going to cross this one off and try again. Does GCU code for alanine?
Yes.
Awesome. So we put our codon line here and we draw an arrow.
AGC.
Does AGC code for serine?
Yes.
Yes.
AAC.
Next we're looking for proline.
No.
Does AAC code for proline? No.
ACC.
So we cross off the A and try again.
Doesn't either.
Does ACC code for proline?
CCC does.
CCC is where
ACC does not code for proline. So we need to cross this one off and try again. When we're looking at CCC, it does code for proline. So we're good there. Does UGA signal for it to stop?
Yes.
UGA does signal for it to stop. So now we've found our chain. The introns then are the parts that we crossed out. So the first intron is AC, because this is the first chunk that we crossed out.
Those are the introns.
And then the second one will be AA. Oh
The
my God.
second intron then
Makes sense.
is AA, this part that we crossed out.
Peter.
The introns are the parts that you crossed out. So this is the part that we need to cut out in order for this chain to code for these amino acids. Yes? You may have to do something like this on the final where you're given an mRNA. Where you're given an mRNA and you're given the amino acid strain and you have to find the introns. You may be given the introns and have to see where they are in the strain. So practicing this process of given the mRNA strain and given the amino acids and seeing where the introns are is a great thing to practice. So continue on with those last couple of practice problems on that page in order to kind of get yourself used to this whole process here. Okay? Once you are done with the practice problems of this, so 7, 8, and 9, you can start on doing the building blocks questions 9 through 15, which we will then grade at the end of class. Okay? All right. I am here if you guys have questions.